From Microbe TV, this is Office Hours. I'm Vincent Racaniello, and joining me tonight from Madison, New Jersey, Brianne Barker. Hello, Brianne.
Hi, it's great to be here. I'm excited to answer questions.
Welcome, everyone. All 124 of you. We'll have more. And I want to thank our moderators tonight. We have Tom and Les and Steph and Vanity Nutrition. I think that's all who are with us tonight. Thank you again for your kind of thankless work. We appreciate it. And folks, while we're here, tell us where you're from. I want to show Brianne all the different places that we can attract here on office hours. So, Brianne, for your classes, do you have office hours?
I do have office hours, yes.
And where do you hold them? In your office?
In my office, generally. Sometimes on Zoom, but mostly in my office.
And
I have a comfortable chair. I have snacks.
Oh, snacks. That's cool.
Yes, there's a drawer in my office with candy in it. And many people come by for the candy from the candy drawer.
Yeah. So you have one office hours per class per course, right?
Total, I have basically 4 office hours a week. I'm teaching 2 classes, so I guess right now that's 2 each.
Right. All right. Well, I conceived of this as office hours for the rest of the world, right?
Exactly.
I also have them for my course. All right. So people responded. This is just great. Here we go. So Kathleen is from New Jersey. Looks like she's got a horse there. That's pretty cool.
Oh, maybe she's close by.
Could be. Rima is from Iowa and Lebanon. Noir, who I know is coming to TWIV 1000, is from Santa Fe, New Mexico. Don't you love that name?
I do.
Black the White.
Yes.
Brian is from Montreal. Paulie is from San Diego. Liam is from Perth, Australia. Dean is from Wisconsin. Pete is from London. Oh, I'm sorry. Yes, you're right. I have Amy under. Let me fix that. Oh, dear. Thank you for pointing that out. I had Amy's label under you. There we go. There's Brianne. Thank you, James, for that. Environmental Coffee House is from New York, Western New York. Frank is from Santa Barbara. Rainey is from North Carolina. John is from Minnesota. Jem is from Virginia. Genetics is from Phoenix. Kang is from Chicago. Kia ora. That could be from New Zealand.
Could be.
Elizabeth is from West Virginia. Carol is from Illinois. And I know Rach is from the Isle of Man, okay, because she's been here a number of times. That's pretty cool, right?
Yeah.
Lewis is from Michigan. Mike is from Ottawa. Remission Biome is from Denver. Ariel is from Westchester. Taos. Ripe DeCamp is from Taos, New Mexico. Steve is from Spokane. Malaysia, that's an interesting one.
That is.
Ontario. Mary is from Boston. And look, I have to show you this one. Brianne is my favorite.
Woohoo!
Nice? PGL is from New Hampshire, Central Pennsylvania, Houston, Texas. I don't know if that's a place. KC. Oh, Kansas City, Missouri.
Kansas City.
Yeah. Here's another moderator, Andrew. Thank you, Andrew, from New Zealand for moderating. Gerringong, Australia. Ooh. How cool. Oh, Tom is at the okay, come on. I'm clicking and nothing is happening. Here we go. Tom is at the Denver airport.
Wow.
Okay, thank you. And Tom is our moderator. Santa Rosa, California, Central Pennsylvania, Northeast Ohio, Ontario, North Carolina, Texas. Brampton, Ontario. Hello, Renzo. How are you? Thailand. That's cool.
That
Chicago,
is very cool.
Southern Turkey. I hope you're okay. I think that's where the earthquakes were, right, in southern Turkey? I guess you're okay because you're here. Calgary, Alberta, Morocco, Queens, Queens is not boring. For some reason, this click doesn't know. Queens is not boring. Queens is an interesting place. Polio Pete. MN is from near D.C. End of Long Island. Boston North. Sao Paulo. Cool. California. New York. South Carolina. Tallahassee, Florida. I'm supposed to go to Tallahassee in a couple of weeks.
Oh.
England, Eastern Massachusetts, Oakland, California, Morocco, we already did that, Utah, Ecuador, Boulder. Oh, and thank you, Kip and Laura, for your contribution. They're from San Francisco, PharmDs. They were at the incubator
I remember meeting them. Yes.
Susan's from Boston, Mexico, Ohio, Seattle, Washington, Chicago, Louisville, Kentucky, Bellevue, Kentucky, and Edmonton and Lewiston, New York. Okay, they're going to be more.
Lots of people I want to visit.
Wayne, Pennsylvania. I'm sure there'll be more. We'll get to them. Okay, let's do some questions. I was telling people at the beginning before we started, I had been going back and forth with a producer for the Tucker Carlson show.
Really?
It was so nasty. He said, Professor,
really?
why did you applaud when Facebook censored all information about SARS-CoV-2 origin? And are you reversing your position now? You know, shit like that, really antagonistic stuff. I don't know why they have to do that.
Yeah, the whole recent situation on that is very confusing. I don't quite understand the motivation behind releasing that information.
Okay, let's see. Here is one from Vanity. We have a lot of immunology questions. After the SC19 vaccine-induced antibodies contract, and we rely on memory cells to respond to infection, is the response all endogenous with cytotoxic T cells and little to no endogenous antibodies? I'm not sure they're both supposed to be endogenous, right?
Right. Yeah. I mean, I would think of them both as endogenous things that we are making. I guess depending on how you talk about it, I could think of somebody trying to come up with some ways to use endogenous and MHC class 1. And in that way, that would work. But in any case, after your cells are your main effector cells are contracting, you're relying on memory cells, both T memory cells and B memory cells. And both of those cell types can respond really fast and differentiate back into effector cells to either make cytotoxic cells or antibody producing cells. So you're not relying totally on T cells. You can have those memory B cells helping as well.
And if the antigen is sufficiently different, then you make new B cells and new T cells, right?
Yeah, exactly. There's been some great data on that that's come out recently.
Yeah. So one of the people, Gabriel Victora.
Exactly.
Right,
that's the one.
Yesterday I was teaching at Rockefeller in a virology course, and Michelle Nussensweig came in for like an hour, and he sat and he listened.
Okay.
And then afterwards, he and Gabriel were sitting outside talking. I'm like, this is the immunology powerhouse.
Oh my gosh! Yeah.
Can you imagine between the two of them, two of the best immunologists in the world?
Yeah, there is some B cell knowledge in that conversation.
Oh my gosh! Yeah, it was really interesting. I wanted to stop and just listen. Right?
Yeah, absolutely.
Okay, Department of Energy report. What did they find, which is classified? So here's what I found. So they say they think it originated from a lab. SARS-CoV-2 came from a lab, but they have low confidence in that.
Mm-hmm. Exactly.
So I emailed a few people, and they said, first, Deborah Birx apparently this morning on CBS said that Los Alamos National Lab virologists were involved, and that is wrong because Bette Korber is not a lab leaker. So that's just the wrong report. Birx is wrong. Well, many people think that they believe the Wuhan CDC was involved, not the Wuhan Institute of Virology. The Wuhan CDC is near the seafood market, all right? However, there's a problem with that. It only opened up in December of 2019, so I don't think they had many samples there.
Right, and were they doing a lot of coronavirus,
I don't think
particularly
they are.
bat corona work?
I don't think they are. I think that's WIV.
Right.
But this puts them in an interesting position because the Department of Energy thinks it may be Wuhan CDC, and the FBI thinks it's the Wuhan Institute of Virology. So it just shows you how screwed up it is. They have no data because both of them are finding different things. And then there's another issue. There's this guy, Xian Jun Hao, a Wuhan CDC investigator who is a bat researcher. And I think they want to blame him. So your question that you just asked, he might be the guy who was doing bat stuff there. And I've read it. There are a lot of articles about him. If you just search his name here, I'm gonna copy it and paste it into the to the chat so people can Google his name. If it's true that the new DOE view is based on Wuhan CDC, then that's going to conflict directly with the FBI report, which focuses on risky research. For bottom line, DOE and FBI may both believe in lab leak, but they appear to be two conflicting lab scenarios. So that's really screwy. I think it's all political, right?
Yeah, I think so too. I think there are other government agencies beyond DOE and FBI that have also released information, some about natural origin, with differing levels of confidence. And so unless we have some idea about actual data, which is not described anywhere, I have seen, in terms of data I've looked at, this idea of this lab leak doesn't make a whole lot of sense to me, thinking about things like the Warobi paper and sequence data.
I tell people that the seafood market was the epicenter. That means that's where the spillover first started. If it came out of the Wuhan Institute of Virology, that would have been the epicenter.
Exactly. And if you look at the genetic evidence of the two different clade A and clade B and sort of when they seem to emerge, that fits with an origin of some animal that was moved into the market.
Right. No, that's all great data, but I think Tucker Carlson doesn't understand it, so he doesn't want to listen to it. But also there are political motivations for saying that China did it. I mean, the guy from Fox News asked me today, do you have money from the NIH which supported communist China research? I mean, what the hell does that have to do with what I have, right?
Yeah, how does money that supports you also support communist China? I'm very confused.
Well, he says he thinks that NIH funded Wuhan directly, but they didn't really.
No.
Anyway, let's do a question. Here we are. Polio Pete, do you think high-risk individuals are more likely to get symptomatic or asymptomatic COVID?
That is hard to exactly address without a whole lot of data. But I think that one of the things that puts many people at high risk, if you think about many of the high-risk conditions, are conditions that tend to make an innate immune response be a little bit lower. So if you think about elderly individuals or some of the other individuals here, we see kind of a reduced or aberrant innate immune response as part of their risk profile. And so in that case, I could imagine perhaps they might be more likely to have symptomatic disease. Although certainly there's also some immunopathology going on. I don't know if we have the data to fully answer that question.
It's a hard one because if the asymptomatic people never get detected, right?
Exactly.
And so you don't know. You'd have to really do a large study where you screened a lot of people and did serology and then asked them, did you ever have symptoms, and then what are your comorbidities, right? So
right, exactly. And you'd need to use a lot of people to get that data.
All right, Susan says, is it possible that post-viral illnesses are caused by intermittent perpetual activation of the innate immune system? Could PRRs be mistaking metabolic byproducts for PAMP damp?
There are plenty of PAMPs or DAMPs that are metabolic byproducts that we know of. So I don't think that part is, an odd idea. I think that perhaps those post-viral illnesses are caused by intermittent activation of the innate immune system. I'm also really curious about whether perhaps aberrant activation of the innate immune system early after infection might lead to some issues with adaptive immunity and a loss of tolerance that can be causing some of those post-viral illnesses. That's something that the data that I've seen and sort of the reasoning that I've seen sounds kind of like it might support.
Peter says, I'll comment on the Wall Street Journal leak. Possibilities aren't facts, and neither are allegations. Speculations aren't theories, and intelligence assets aren't witnesses. They should stick to chasing UFOs. That's very well put. I like that. All right, Simon. I'd love to hear how much we know and don't know about the difference in the immune system of old people versus adults versus children versus babies and between men and women. Oh, yeah.
I think that there are a lot of great questions there. So one piece to all of this has to do with the amount of immune experience that someone has in terms of how many memory cells and how much cross-reactive memory an individual might have. So one big piece is just going to be about immune experience. Individuals who are older are going to have more memory than individuals who are younger. We seem to see that older individuals seem to make a little bit less of the innate immune response. And in some cases, kids make a little bit of a different innate immune response as well. Exactly why that is for children, we're not really sure. I've seen some great data sort of coming out from conferences about some of the data on older individuals. There's also some idea about this thing called inflammaging, where sort of increased damage in different cells can change innate immune responses in older individuals, as you're acquiring different kinds of damage as you age. The difference between men and women is really fascinating. So Sabra Klein's lab has done a lot of work on the role of different hormones on immune cells and has shown quite a few differences. The general idea is that women seem to have a somewhat increased or higher immune response and maybe more prone to immunopathology, where men have a little bit of a lower response and are prone to more pathogenesis or direct damage from infection. Some of this is also due to a bunch of immune genes being on the X chromosome and escaping inactivation, so that they are actually different in terms of gene dose in men and women. And that's true of some of the TLRs like TLR7 as well. So I think that there's a lot more for us to know. And these are some topics that people are just now starting to really address. But it's fascinating.
Oh, so someone asked for you to define PAMP and DAMP.
Okay. So a PAMP is a pathogen-associated molecular pattern. So it is a molecule from a microbe that is unique to a microbe and not to our cells. And this is a target that the innate immune system can recognize. Some people also call it a MAMP, a microbe-associated molecular pattern, because the pathogen the microbe may or may not be a pathogen that's making this. We've also realized that there are some molecules that are made by our own cells when those cells are damaged that can similarly induce innate immune responses, and those are DAMPs, damage-associated molecular patterns.
Holio Pete says, why are the bivalent boosters under EUA when the primary series is fully authorized?
I think that that's a timing thing and a question of the amount of data that we have on them.
Yeah, they're relatively recent, right? So I need to get more data. Okay. Kang says, does adaptive immunity senesce at a faster rate than innate immunity, since deaths from COVID are due to the effects of a cytokine storm?
That is a great question. I I don't have a total answer for you, but I will say that the adaptive immune cells are much longer lived than many of the innate immune cells, which are going to be replaced more often. And so I would worry more about the senescence of the adaptive immune cells, since those cells are living for a long time and the innate immune cells are getting replaced. And so if a cell were to senesce, it would be it would be replaced relatively quickly. But I'm not sure if I know the comparative rates of those two.
Are all cytokines produced from innate responses?
Oh, no.
Or some are later, right? Yeah.
Yeah. Cytokines are just basically the messenger proteins in the immune system. They are produced by adaptive immune cells, innate immune cells, and non-immune cells.
Right. So that's because Kang thinks that since you have this cytokine storm, it must be innate responses, but it doesn't have to be. It could be, could be both. Yeah. All right. Why do some patients with severe infections have pancytopenia and neutrophils are very, very low? Then a few days later, the counts go high.
So I think this would be related to some of the cytokines that are being made that might either induce neutrophil production from the bone marrow or otherwise induce neutrophil recruitment.
Nicola is in Italy. Good to see you again. Hello, Brianne. What's more responsible for preventing severe disease and death? Neutralizing antibodies or cellular immunity? If cellular immunity, how can boosters be justified?
That's a great question. I think that there are people who would argue either side of antibodies or cellular immunity. I think there's a lot of data on T cells and adaptive immunity being really important in preventing severe disease and death. But I don't know if we are totally certain on that. I think that I would love to see some more data. I've seen some from Daniela Weisskopf about boosters and how they influence T cells, but in theory, they should influence T cells, but her data didn't show that influence being as much as I would have expected. Love to know. So,
first of all, part of the they don't have to be neutralizing, right? They can be non-neutralizing, FC mediated. So there's that. But also, when you get a booster, as Paul Offit said, you get a boost in antibodies for a couple of months, and that protects you for a while, right?
Right. So, there is sort of this protection from getting infected versus protection from later disease. Yeah. And so getting protected from infection does help you pretty well against severe disease too.
Yes, it does. The thing is that if you do get infected and you have been multiply vaccinated, you'll probably be all right unless you are really, really old or have comorbidities, right? And those are the people who can die, and those are the people who you have to have a Paxlovid plan or something similar.
Right. And so, but preventing infection in them is going to also be particularly important.
Yeah, Paul Offit said that these people who have issues, just the slightest infection pushes them over, right? So you don't even want them to be infected at all. Yeah. John wants to know, what are the major differences between immunology and virology? Yeah, you got them right here.
Yeah, I mean, with immunology, we're thinking about sort of the host side. I kind of think about this as I'm thinking about two different sides of the battle with the immune response and viruses. There are lots of immunologists that think about things other than viruses in terms of autoimmunity or immune system development. And virology is, really more thinking about viruses and their impacts. I put the two of them together myself and think of, immunology as being a way that we think about viral pathogenesis.
Yeah.
And so to me, that's where I see the intersection, but there are lots and lots and lots of intersections, even going to places like the idea that these really important immune genes, RAG1 and RAG2, may have been inserted into our genome from a transposon that may be related to a virus way in the past. So they're closely linked.
So there's a lot of overlap, but there's also things you do in virology. You study nucleic acid biosynthesis in cells. And it doesn't have much to do with immunology, and developmental immunologists can do things that don't have to do with responding to antigens, right? But there's a
Right.
lot of overlap for sure.
In fact, I was sort of an outlier in my graduate class, in my graduate programs. Most of the other students were really excited about really autoimmunity questions, T cell development, things like that. And I was the outlier who wanted a pathogen.
Yep. Yeah, but now that we've had a pandemic, many immunologists are pulled into the pathogen fold, right?
Yes, they are.
Here's a note for you from one of our moderators. Thank you, Brianne, for your internet presence of deep, broad knowledge and good humor. Kind heart, too. Love your cat.
I don't know where she is. She may make an appearance at some point. Thanks, Tom.
Where did you get this? Mice, lion, monkeys exaggerate. I don't know. Alan Dove said it years ago on Twiv. I don't know where he got it. I mean, if you Google it, you will find, I think, not the exact thing. So I'm not sure, but I like the way we were using it. Okay, these are all the people telling me where they're from. We'll have to scroll through that. Okay, here we go. Do we know what exactly proteins are presented in germinal centers for positive and negative selection of T cells? Like, what organ do they come from?
I think the idea in the germinal center, well, so I guess there are a couple of things going on here. So positive and negative selection of T cells happens in the thymus. The germinal center is really a place where we see some selection for B cells happening. So the antigens that are present in germinal centers that lead to positive and negative selection of B cells could come from anywhere because they could flow into the lymph node from the lymph. So really they could be secreted proteins or proteins as a result of damage from any organ. The proteins for T cells are being transcribed and translated by medullary thymic epithelial cells, which have some really unique ways of doing transcription. So they can actually pretty much transcribe all antigens. There are some that are brought in by some other antigen presenting cells, but the MTECs, the medullary thymic epithelial cells, are pretty important.
While you have a moment, folks, hit the like button, please. We have 340 of you, 160 likes. Let's make it a little even there, please. It helps us attract more people. For Los Comments wants to know what you think about a reasonable boosting frequency for COVID-19 vaccines would be, in the extent to which imprinting should be considered.
So I think that there's a lot that we are still learning about imprinting. I think it's hard to fully know about the antigenic distance that we're going to see between different viruses and future viruses to know about how we put imprinting in. I think there's still a lot that we're learning about imprinting. And so that is a tough one to answer. I guess the question with the reasonable boosting frequency is, what exactly are we trying to prevent? Are we trying to prevent severe disease? In that case, we've seen T cells, at least in the data from the Weisskopf lab, seem to really be sort of maxed out with a first dose of a vaccine. And we need to see how long they persist. And that would really give us some information about a boosting frequency. If we're thinking more in individuals about preventing infection, then we need to be boosting more frequently to bump those neutralizing antibodies up. So the question first, I guess, is to come back is, what is your goal with those vaccines?
Yeah, I mean, I had 3 doses, the original 2 plus a boost. I didn't have a bivalent. And when I got COVID in November, I just took Paxlovid and it was over in a day because I have no comorbidities. I'm 70, so I'm not quite at the elderly. But even then, I think I would just stay with these 3 unless the virus changes substantially, which I don't see particularly happening. But, if you have issues, then maybe you might want to boost every year. I just don't know.
Maybe if there's some data that the immune response, fades at 10 years, in 10 years you might want
Yeah,
a
sure.
booster.
I think it was cool what Daniela said. We can't tell how long T cell memory lasts because people are just getting infected every year and
I,
boosted, right?
I know, exactly. No, very true. And I don't know how much, if people have seen this. There's a paper that came out relatively recently about T cell memory in mice, where they tried to actually see how long
Yeah.
memory T cells
Yeah.
could live and could persist. And they were taking cells from one mouse and transferring them into other mice. The lifespan of a mouse is 3 years, and they actually saw they could get the memory T cells to live for 10 years. So in that paper, at least, it indicated that memory T cells live longer than the lifespan of the host.
Wow.
So they may live for a really long time.
Wow. That's a long
Yeah.
experiment.
Yeah, exactly. Exactly.
Gosh, I wonder if they had NIH funding for that, right?
They
Because
did. It's a group that has been doing stuff like this for quite a while.
you have to have faith because it's a long-term thing. It's interesting.
It is.
Patricia wants to know if you've ever read Commotion in the Blood, and what do you think if you have?
I have not ever read Commotion in the Blood. I'm going to have to look into that.
All right. Claire says, for unvaccinated, COVID-naive, healthy population, if you get infected, antibody higher versus the first mRNA shot. So is antibody higher versus the first mRNA shot? Will this mean better to wait longer interval for the first mRNA shot between the two shots?
So if you look at the data on this, there are some people when they're infected that make a higher antibody level than the first mRNA shot, but there are also many who do not. There's much more variation with those antibody levels on infection compared to what we see in the mRNA boost. So sort of upon infection, among many other reasons why infection is not ideal, you're sort of gambling on what those antibody levels will be compared to the first shot. I think that there definitely is some data that maybe the original amount of time on the interval for the two shots was less than ideal, and spacing that out did seem to give a slightly better response in terms of antibodies. And I don't know if I've seen all of the T cell data on that.
Is the main cause of mortality human internal health related to such as heart disease, cancer, stroke, diabetes, all tied back to the immune system failing?
So I have said to my students before that if you look at the top 10 causes of death in the world, 9 of the 10 can be related back to immune responses, and particularly innate immune and inflammatory responses. And as I tell them, someday I'll figure out a way to tie in traffic accidents too. But there are many causes of mortality that have a role for the immune system. Whether the immune system is the number one contributor or not, I don't know if we can totally say that, but the immune system can contribute.
So if you look at people with B or T cell defects, you could kind of start to address that, right?
You can definitely start to address that. But you can also see that if you are using immune modification, many of these disease states can be impacted.
Yeah. Tuj also wants to know why certain vets develop Gulf War syndrome.
I am not sure I know anything about that. Great question.
All right, Carol, our nephrologist who answered a question for us a couple of weeks ago. Well, perfect SAT, wow. Several reports describing cases of ANCA-associated vasculitis after COVID-19 mechanism.
I think that there are some data on differences in antibody production following vaccination versus infection, particularly with relationship to fucosylation of the antibodies, and how much of a sort of self-reactive antibody response comes up in infection because of the cytokine storm impacting B cells that should have been turned off. So I could imagine that as being the mechanism. But I am not sure that I can tell you for sure that that's the mechanism.
Okay. For vaccine hesitant groups, could 2 mRNA shots with an interval of 6 months be a good option? Trying to encourage a family to get 3 shots, but 2 might work more easily on them.
2 mRNA shots with an interval of 6 months is 100% better than 0 mRNA shots.
For sure. Yeah.
To me, the interval 6 months seems a little long, but I don't have an answer for what the perfect interval is, and you need to balance how long people are going to be staying unprotected. So if they are still hesitant at this point, then 2 mRNA shots with an interval of 6 months is certainly better than not being vaccinated at all.
Yeah, we used to talk about this early on, where the 2 shots initially a couple weeks apart were too close, right, to get proper maturation. So
6 months seems a little long, though.
I know in the UK they did some trials, maybe not 6 months, maybe shorter, but
I'm thinking more like 3 or 4 months. Yeah.
Okay. Brianne, can you explain how an FC portion can work while the neutralizing portion does not? Sorry, I'm immunology novice.
Sure. So if you think about an antibody, you can think about an antibody as being kind of shaped like a Y. My microphone's in the way. So we've got our Y-shaped antibody. The FC portion is that sort of stalk, the tail of the Y. And some of the ways that antibodies act is that will be that they can, that tail of the Y will bind to different kinds of receptors on other immune cells and induce those immune cells to make responses and induce those immune cells to do things. When we talk about neutralization, we're specifically talking about the antibody binding to a virus, in this case, and that's physically blocking the virus from interacting with its receptor. And so that could just have to do with those little shorter parts of the Y, not the longer FC portion. The FC portion could be involved in that blocking, but it doesn't have to be. But for the other things, the FC portion needs to interact with some kind of receptor or some other kind of protein.
Richard wants to know, all right, so the FDA panel recommended 2 RSV vaccines for adults. Yes, very interesting. So Daniel talked about these last week. They're 2 New England Journal articles. One is an F protein vaccine, so it's an F protein, purified protein, and the other is an adenovirus vectored prefusion F protein vaccine. So one is vectored, one is protein, and they both look good. They have good protection against severe disease in adults. And he also said that Pfizer is coming out with a vaccine for pregnant women, which will allow the antibodies to go to the fetus, right? So you immunize the moms instead of
that's fantastic.
Baby. So that's very exciting there. And by the way, did you hear that Novavax the company is going bankrupt?
I did not. I've sort of been wondering what happened with them and where and when we might see some of their vaccine. But that's too bad to hear.
Yes, so Bloomberg said Novavax stock tumbles after warning of doubt over the future. They don't know if they can stay in business another year.
Yikes.
Because they're hemorrhaging money.
Right.
They never got to sell a whole lot of vaccines, sadly.
I know. That's too bad. I'm sure that there are many people who would have been excited to take that vaccine.
Well, many people who are scared of mRNA vaccines, right?
Exactly,
They
yeah.
like that. Okay. The lab leakers never provide any phylogenetic trees. Of course not. They don't have any data. It's all
That is
innuendo,
correct.
right? There's no trees. Yes, and the other side, which says this is a zoonosis, has plenty of trees. And they're lovely trees, yeah. Oh, look at this. We have a teacher here who's saying to her students, let me know via Canvas if you're on tonight. So they're watching you, Brianne. I don't know what
Hi, students.
what Tia is teaching, but that's pretty cool that they have to watch you for extra credit. Brianne, how do vaccines help FC-mediated antibody effector functions work? If it's too complicated, cover an immune or TWIV.
So vaccines can help us induce more antibodies, so just a higher number of antibodies. And they can also help us make memory B cells to make those antibodies over a longer term. But every B cell starts out making its antibodies with an IgM tail. Some of those functions can be done well by IgM, but sometimes the antibody needs to switch to use another isotype, another kind of tail to be able to do some of these other effector functions well. And you don't get that until a little bit later in the response. And so a vaccine can also push your B cells to class switch so that they're class switched before you get infected. And you already have the type of antibody that makes that FC mediated function work.
Glycosylation is important for FC function, right?
It is, especially the fucosylation or afucosylation. Right? Yeah.
Yeah, we did a paper not too long ago on TWIV.
That
right?
paper is really fascinating.
Very cool. All right, Wanda said, I did read that the February edition of the prestigious medical journal that natural immunity is just as effective as 2 doses of Moderna and Pfizer. Is this true?
From what I've seen, there's a bigger range with natural immunity. So you don't know that you're necessarily getting the immunity as reproducibly. I've seen differences in fucosylation of the antibodies, like we just mentioned before, coming from natural immunity versus vaccines. And so some of those antibodies that are made by the vaccines may be more effective. It's really a question of what you're measuring in terms of 2 doses of Moderna and Pfizer. And there also doesn't seem to be some of the risks of, say, long COVID or some of the other risks associated with
how about dying,
risk of dying also coming from infection.
Yeah, I mean, that's the problem. Is that with a vaccine, you have a head start on your immune response, right?
Exactly.
So Nevitz heard a webinar today on the development of suicide substrates against the helicase by Taran Kapoor. Wonderful story. Would be nice to have him on or discuss the paper. And the paper is force-dependent stimulation of RNA unwinding by the helicase. So this is a kind of stuff that it doesn't go on much with SARS-CoV-2, right? Because it's mostly vaccines and antibodies and T cells and so forth. So I'm glad to see people are doing. I'll have a look at it. Thank you for that. Is there anything on the bird flu horizon that you find noteworthy? Yeah, nothing, right?
Yeah, I mean, the human case in Cambodia was interesting, although with the data that's come out since, it seems like that's an older strain. And I haven't seen any additional data that seemed really noteworthy, but it is interesting that it's in some mammals. It's interesting that there's spread with so many birds, and that every infection gives more opportunity for mutations. So we should keep an eye on it, but nothing terribly new.
Yeah. So Spamlet thinks I should go on the problem with Jon Stewart. Okay, you guys all write him and tell him to get me, and that'll get me on. That's what got me on Lex Friedman. You guys writing in. All right. No, sorry, that's to Polio Pete. Ideally, what would you like to see your students end up as? Meaning, how would you like for them to use what they have learned in your classes?
Great question. I think I would like to see my students as people who critically think and use data and evidence really well and are in a job that they like. So I get really excited when my students, go into medical school or graduate school or into pharmaceutical industry or into teaching or into all sorts of different things, as long as they are going to use evidence and think critically.
That's a good answer. Do something that you're passionate about, right?
Yeah, they sometimes will come up and they'll be like, "Oh, I decided to go this route. Are you disappointed?" And I'm like, "Are you happy with that route?" And they say yes, and then I'm like, "I'm happy with that route too." Um, and that
Yeah.
was how I know that my mentors felt about my path. That was the key thing my PhD advisor asked when I started teaching was, "Are you happy?" And I said yes, and he's like, "Then that's what I want you to do."
Alan Duff has told the story. He came in my office. He said, I'm going to go into science writing." And I said, "I think that's great."
Yeah. Yeah. Well, so when I told my PhD advisor I was going to start at teaching at a small liberal arts college, his response was, "Well, of course you are." And I had been like really, really thinking about this, and it had been really, difficult for me. And everyone else who I talked to when I made the decision was like, "Well, yeah, that's exactly you. Of course, that's what you're doing." So I was like, "Why couldn't you guys have told me this before?"
All right. Well, we don't have an answer for this, but can you explain why avian flu is able to infect humans from animals but not between humans? Well, that's the $1,000,000 question, right?
That is the $1,000,000 question. Perhaps the dose that humans are excreting is not high enough to infect, or perhaps the virus is not getting to a place where it can leave humans for transmission well. Or
You
maybe
remember the ferret studies where they adapted it to airborne transmission among ferrets. There were a number of changes in the HA that affects the stability, but maybe those are not
right.
compatible with bird transmission, which is the major route.
Right. I mean, there are differences in sialic acids, differences in temperature in birds. So,
and whether it will happen, who knows, right? That's the key,
yeah.
Anders. We just don't know this. Let's see. Well, here. Okay. Flu. Do you think it's possible to find a universal neutralizing monoclonal antibody for SARS-CoV-2, all variants?
So I think that there have been a couple of sort of universal SARS or universally coronavirus vaccines out there, haven't there?
People are working on them, right?
Right. People are working on them. I don't know if those are neutralizing antibody based or not. So I don't know. We'd have to really find the right epitope, and we'd have to figure out a way to make that epitope in a stable manner. And I am not a structural person.
Yeah, I just don't know. I mean, there are broadly neutralizing HIV antibodies, right?
Yes.
That hit all the variants that arise in a patient, right? But you
Yes,
actually need
absolutely.
more than one. You need more than one to cover them all, right?
Yeah, you need a few.
And I think that's a really challenging problem. So,
yep.
I think it's possible, but whether it'll happen, it may be very hard. I just don't know, right?
Right.
Okay, this thing keeps jumping up and down. I don't know why, but here we go. Do we know how many people died from using ivermectin to treat COVID? I have no idea.
I don't know.
But did you see now they want to treat flu and RSV with it? They want to treat everything. There was an article about doctors who are prescribing it for flu and for RS infections.
Why?
Because apparently it works against everything. And apparently it's being, hidden by the big pharma who want to make money on other things. That's just all nonsense.
Yeah. I don't get it.
It's crazy. All right. How does MHC influence these age and gender differences that we talked about before?
Yes. So MHC sort of makes all of this harder, particularly in the T cell realm, to study because each person's MHC is different. We have a combination of I usually say 6 MHC class 1s, 6 MHC class 2s, and they are all different from other people because there are so many options. And so they don't change due to gender or age, but they mean that each person, depending on their gender or their age, has this other big variable that drives differences in the immune system. And so you can't look at just two people of a particular gender or two people of a particular age. You have to do huge studies to really get at this because of all the MHC variation that's going on in there too.
Okay. This is weird. It never jumps like this before. Okay, we had our neutralizing, we had COVID, we had MHC, and here we go. Is someone who was vaccinated then infected with BA2, would their B and T cells be able to recognize the current variants more readily since they are recombinants of BA2? You get every possible question here. It's just amazing.
Yeah, I
right?
see that.
So BA2, since the current variant, so they're talking about later than BA2 here, correct?
Mm-hmm. They are. I don't think that there would be a huge difference given the broadening that happens as the immune response goes on. I think that the immune response is hitting a pretty large number of variants as it broadens, but I am not sure.
Yeah, I don't know either. Sorry about that. Okay. Okay, here we go. There's a Paul Offit question here. Where does this 2 months thing come from, Dr. Offit? You get a boost for a couple of months in antibodies, basically, right?
Yeah. So there was data from Israel, I think, pretty early after one of the boosters was described that sort of showed that there was an actual change in how frequently people were infected for about, I believe it was 7 weeks was my recollection of that data from Israel. And so I think that that's where the 2 months is coming from, is that you have this 2-month antibody boost that protects from infection before your protection goes back down to sort of baseline.
Okay. I don't know. This is weird. Do ketone bodies affect the turning on of adaptive immunity after acute infection?
I do not know much about ketone bodies, so I couldn't tell you.
Yep, and I certainly don't. How about any new data on persistent oral lichen planus and the CD8 T cell response aberrations?
Yeah, not too sure about
that one either.
No.
Okay. How does the immune system vary throughout the day and night? There you go.
There's a paper that just came out about that. It does seem like there's some circadian differences in the immune system, though people have studied it relatively little. And it's hard because most of these immune responses are studied in mice who have a different circadian clock than do humans. But there was a paper that came out, I think last week, indicating that the immune response to vaccines differed if someone was vaccinated at different times of day or night. And I think they looked at 3 vaccines, if I remember correctly, and they actually saw different times of day was better for each of the vaccines. So I know there's that one paper that just came out, and wow. That's all I really know that's been done in human.
So when should we get vaccinated? What time of day?
Which vaccine are you getting?
SARS, mRNA vaccines, say.
I'd have to look back at their data. I think that one was morning.
That's funny. All right. I wanted to ask your thoughts on the recent Nature paper that posits the concept of atherosclerosis as a bona fide T-cell autoimmune disease targeting the arterial wall.
I think that atherosclerosis, along with a lot of other disease states, can be a little heterogeneous. And I think that there probably are some cases where T cells are attacking the arterial wall, and there are probably some cases where it's not.
Yeah, it's never 100%, is it?
No.
All right. Is there an immunology book that's between Immune by Kyrgyzak and the big immunology tomes? I enjoy Immune, and I'd love to dig a bit deeper before buying a 1,000-page book.
Yeah, I mean, I use the PARM book, The Immune System, with my students, um, which is sort of written more at a level for undergraduates. There's also Kubi um, Immunology. So those are the two that I use. I don't know which books you mean by the big immunology tomes, but they're definitely both books that are under 1,000 pages. So I would go with those, but I think there is definitely a lack of some immunology info out there.
David wants to know, does MHC1 on a virally infected cell present self, viral, or both at the same time? How does this work?
Yeah, so every virally infected cell probably has many different MHC class 1s on its surface. So I don't know, we'll imagine 1,000 MHC class 1 molecules on the surface of the cell. I just made that number up. Some of them are presenting self-antigen. Some of them are presenting viral antigen. And if there were just self-antigens being presented, there would be less likely to be any co-stimulatory molecules made by that cell to turn on T cells. Most of those co-stimulatory molecules are made because of the PAMPs and DAMPs that we mentioned earlier. And many of the T cells that could respond to those self-antigens were either eliminated in the thymus or have been turned off by tolerance mechanisms. So we've got MHC presenting all sorts of peptides. The MHC doesn't know where the peptide came from when it's putting that peptide on the surface. And then a lot of the discriminations happening on the side of the T cell. The viral antigens have T cells able to respond to them and T cells that can get the co-stim. The ones that are self-antigen being presented might never be seen by a T cell because that T cell died in the thymus.
Right. Tia wants to know, can you talk a little about how viral DNA can be co-opted and reused as elements within our own DNA? For example, fetal protection from ERVs.
Yeah, Vincent is definitely one who also can talk about this virology part pretty well. But there are certain viruses, the retroviruses, that have an enzyme called integrase. So they are able to integrate into the genomic DNA of the cell they infect. And if that cell lives for a long time with that integrated DNA, the DNA might be able to acquire some mutations, have some parts get deleted. And if it just so happens that that is a cell that will then transmit to the next generation, so some kind of germ cell that the virus had infected and that we've integrated into, then the DNA can go to the next generation and maybe be part of selection, natural selection, and be co-opted and reused like the syncytians involved in fetuses and the placenta.
Yeah, and some of these ERVs are transcribed at the single and double and 4-cell stage of embryos, and they induced some ISGs, which are thought to be protective of the fetus against infection. It's very cool. Can the innate immune system, get stuck on without persistent DAMP, PAMP, etc.? Stuck on, yeah.
I don't know if it can get stuck on without PAMPs and DAMPs, but I think that sometimes we underestimate some of the DAMPs that our cells are making, and that may lead to some of that persistent activation.
Genetics, thank you for your contribution to science education. We really appreciate it. Lori wants you to explain the mechanism for neutrophil lymphocyte ratio being an indicator for COVID severity.
I don't know this for certain. I could imagine that we see a lot of lymphocytes because of a different profile of innate immune cytokines. And so if you are making the cytokines that are bringing in neutrophils or inducing neutrophils, perhaps you also are making cytokines that are going to give you a more severe disease. But I'm speculating there. I don't have any actual data on that.
Is that MN says the cat that I'd see on the back of the couch during the earlier days of the pandemic, right? Athena.
Yes, she's
right?
Athena is actually on the couch right now, sound asleep.
By the way, folks, if you would like to go to TWIV 1000, you better get moving here because tickets are going fast. You have to send me an email, vincent@microbe.tv with TWIV 1000 in the subject and say you want a ticket. And if we have one, we'll get it for you. All right. And then we suggest a $100 donation. I'll let you know how you can do that. But this is probably your last chance this week. I have a feeling they'll be all gone. We only have 130 seats, and people seem to want to go. It's pretty cool. See all of us together. Thomas says, my first true exposure to the complexities of immunology was in grad school in a course titled Neuroimmunology. Hats off to the immunologists out there. Neuroimmunology.
Yeah, that sounds like a complicated place to start.
Right. Thank you, Kathy, for your contribution to science communication. We appreciate your support. All right, Stephen said, when I got COVID, I had some stomach issues. I was wondering why some people get stomach issues. Is that from the virus infecting cells in the gut?
What do you think about that?
I think it's all cytokines, frankly. I think with flu, you get stomach ills. Flu doesn't replicate in your stomach. I think it's a cytokine effect. Does that make sense?
That does make sense. And I was going to say, I would say cytokines are at least part of it.
So what else besides a cytokine would do it? I mean, would cells infiltrating in make your stomach hurt? Probably not, right?
No, I have a hard time imagining cells infiltrating into the GI tract if there is no virus there.
Yeah, it's probably cytokines then. All right. That's what we think, Stephen. Of course, someday people might find some virus, but I have not seen it.
Yeah, I don't know.
I mean, with SARS-CoV-2, there's some papers where they see a particle here and a particle there in the intestines, right? And they say, okay, it's replicating, but
how do you know it's replicating?
How do you know it's replicating? And there's so few. What is that doing? I think you have to be very careful about all that. Okay, here's Neva from Buda. Great info on convalescent plasma. What do you think, Brianne? You weren't on that episode, right? Did you?
I was not on that episode. I've heard some of it. I haven't actually finished it yet. So I think that convalescent plasma is, something we've used for other infectious disease for a really long time, and it can be really useful. My biggest worry has always been about what else might be in that plasma, that convalescent plasma. But I think that, everything in our arsenal is important to helping out these patients.
So Arturo said, for immunocompromised patients, it's very useful.
Which makes sense.
And he said that convalescent sera from a person who's been infected and vaccinated neutralizes all the Omicron subvariants. All right. Mm-hmm. So he said it's not for most people, because most people now have high immunity because they've been vaccinated. But for the immunosuppressed, and he also said if we have a H5N1 pandemic, that's what we should use right away before we have a vaccine.
Right. That's the thing, is that there are certainly situations where this is a key tool in the arsenal.
Here's our other moderator, Peak. Welcome. Peak is a high school student, Brianne.
Oh, hi, Peak.
He is over on he's a mod over on the Discord server. I don't know, maybe you've seen him there. But he helps us out now and then. All right, Patricia, does stress decrease immune function? Because cortisol can suppress inflammatory and immune responses and can decrease circulating leukocytes and inhibit production of certain cytokines.
Yes. And in fact, if you had just asked me whether stress decreased immune function, I would point out that a lot of it is because of the effect of cortisol on inflammatory and immune responses, and all of the things that you point out here. So yeah, absolutely.
What was Which one was this? Patricia. Okay. Where did it go? Sorry. Okay, here it is. What is immune profiling?
So immune profiling is basically trying to broadly characterize an immune response or the immune system of an individual using things like thinking about transcripts or thinking about a lot of different cytokine proteins instead of focusing on just one individual molecule or cell type.
Okay, from Claire, 61-year-old male smoker, COPD, unvaccinated, got infected, recovered, COPD got worse. How soon is the best timing for the first mRNA shot?
I believe this is a Daniel question.
It is, yes. It is. It is a Daniel question.
Yeah.
I don't know what Daniel would say, but I suspect he would say when you have stabilized, right?
Yeah, I
Most
think
likely.
that when you're feeling well, when you're healthier, I would say that's probably the best time.
But if you want to ask Daniel, Claire, Vincent at microbe.tv, and we're recording tomorrow, so you should zip it over tonight, and I'll put it in the queue. Robert, thank you for your contribution to science communication. Really appreciate it. Environmental thinks you're probably an amazing professor. Anyone taking a class has got to be the luckiest person.
Thank you. Can you tell my students to write that on the day they do their evals?
Yeah, evaluations, right. Vanity thinks of antibodies as an airplane. The two ends are wings and the stalk is first class. That's very good. I like that very much. Okay. Office hours. Okay, Lisa, thank you for your contribution. Really appreciate it. Wasn't Novavax the company that does the shingles protein vaccine? Shingles? No, that's GlaxoSmithKline. That's Shingrix is GlaxoSmithKline. And Novavax, they have one product which is the Novavax COVID vaccine. And unfortunately, it doesn't sound like we're going to have it around for much longer. It's too bad. Brian wants to know, why don't we develop lasting immunity against C. diff?
Awesome question. I think that there are a lot of immunologists and C. diff individuals who'd love to know the answer to that question, and I do not know the answer.
How would you explain to an individual with little science background that SARS-CoV-2 is more likely to have originated from nature rather than a lab leak?
I think that I would say that SARS-CoV-2 emerging from nature fits really well with a pattern that we've seen with the emergence of other infectious diseases in terms of the presence of similar pathogens in animals, and then sort of the spread around the world. So a lot of the factors that we think about for how HIV emerged or how many other infectious disease emerged from nature also can see parallels with SARS-CoV-2. And we don't have other examples, and the data doesn't seem to fit some of the lab leak scenarios as well.
Yeah, I like that. And I also would add that there is a lot of evidence that the virus came from nature and zero evidence that it came from a lab. Zero, -0, if that could be. It's just no evidence.
Yeah, and if you're going to think of something that's so out of the rest of the patterns, then you need extraordinary evidence, not zero evidence.
The SARS accessory proteins, all these, are considered interferon antagonists, and it can impair the host's immune response through various mechanisms. Okay, Peter is making a statement.
So that's cool. Correct.
That's very cool. How are new vaccine adjuvants introduced? Phase 1, 2, 3 trials, some other way?
Yeah, phase 1, 2, 3 trials. And there's a lot of looking at the quality of the of the immune response, as well as the side effects. And you can imagine that if you are making an adjuvant that's trying to induce an innate immune response, you may get some fever and some malaise and things like that. And thinking about that trade-off on adjuvants has been a big issue in adjuvant development.
Yep. All right, so Tom writes, with Gulf War syndrome. So Gulf War syndrome is this multi-symptom disorder, right? Kind of like CFS, kind of like long COVID, but multi-symptom,
Right.
unexplained. And he says there have been many, many hypotheses, but no consistent data indicating a mechanism. And I think that's a fair summary of it. Yeah, so genetics, what is Gulf War syndrome? That's it. Like fatigue, headaches, joint pain, indigestion, insomnia, dizziness, all that stuff. These individuals develop that, and nobody knows why. Yes, we read about another person cured of HIV. What is behind these headlines, and what might this mean with respect to future HIV treatment protocols?
So for a lot of these individuals who have been cured, the most famous of whom is the Berlin patient, but there have been some that are more recent. What has happened has that these people have had a bone marrow transplant after some kind of treatment, like a chemotherapy treatment that basically wiped out all of their immune cells. And I guess we'll say wiped out and all of their immune cells. And then they got a bone marrow transplant with bone marrow that is from an individual who has a mutation in CCR5, so that those new immune cells that come from that bone marrow can't be infected. I think that this is awesome for those individuals. I think that there's always this question in the field of, did you wipe out all of the immune cells? Is there a little bit of latent virus infecting some cells that escaped depletion? But I also think that it's difficult to imagine how 35 million people in the world can be cured by this, both in terms of we can't give bone marrow transplants and we can't do chemo on all 35 million people who have this. I think that it tells us the importance of CCR5 and using some of our CCR5 directed therapies that we already have. And I think it's awesome for these people. But I don't know how much is going to change future HIV treatment protocols.
Yes, Pamela Bjorkman. Remember, she was making that broadly active COVID vaccine. Remember, she talked about it at ASV. So that's one that could potentially neutralize all variants. It's not a monoclonal, but it's a universal COVID vaccine potentially, right?
So if you had that universal, maybe you could then find an antibody and make it as a monoclonal.
Right. Thank you, Silvio Pina, for your contribution to science communication. Claire writes, when you said there is a bigger range of immunity from natural infection versus vaccination, are there any common features from those who do not get as good immunity from the former?
I think that all that we know are about, older individuals or individuals with other sort of comorbidities that might impact their immune response. But in terms of some of the other things that impact the range, I'm not sure that we do know. You'd need a huge study to really tease that out.
By the way, we recently lost a giant in vaccine RD, Emil Gottschlich from RockU. Yeah, he developed the first meningitis vaccine. That's right. He just died on February 14th. I didn't catch that. Thank you for pointing that out. All right. What's the worst case a bad COVID vaccine could do? Just not mount a response, like due to spoilage, wrong dosage, improper processes? Just curious. What's the worst case a bad vaccine could do? I would guess not to mount any response at all, right?
Yeah, probably not giving you much of a response. I mean, when you say improper processes, I don't know if that would include a bad vaccine delivery. I think that there can be some errors sometimes in injection that might cause some damage in the arm. But that's not really the vaccine being bad. That's a bad delivery.
You know, so, when you immunize many people, some simply don't respond, and you don't know it
Yeah,
because
of
you don't
course.
check, and then they become ill because that's the way it works on a population basis, even with a perfectly
Right.
good vaccine, right? So,
Absolutely.
but I used to think, what if they didn't put vaccine in hundreds of thousands of vials, right? You could imagine that might happen in a production facility, but
Sure. But I think that they have an awful lot of QC
they should.
measured to make sure that doesn't
Yeah.
happen.
Yeah, I mean, I'm sure I'm not aware of what they do, but
No.
yeah, so that's where fears come from when you don't know. So I'm not afraid. I'm not worried about not getting vaccine. Some elderly still have allergies. Does this mean their immune system is not senescing?
I don't think so, because those allergies might be the response of a couple of different, B or T cells responding to the allergen. That doesn't mean that other B or T cells aren't being impacted. So I don't think that any part of the senescing is a all or nothing kind of thing.
My tonsils were removed 60 plus years ago when I was little. How would my immune system be different if that hadn't happened?
You would have one more secondary lymphoid organ and one more place where you could make immune responses. But I don't think that your lymph is not getting filtered or not getting surveilled because of the loss of just those tonsils.
Andrew tells us that pinworms know the difference between night and day, and when they're in the body, how they do it is not clear. Okay.
Very cool.
Has there been any more information from FDA and CDC about trying to make the vaccine an annual shot? No, they're thinking about it, right? We don't know.
Yeah, I haven't seen anything specific about it.
What are your thoughts?
I'd want to see data on what kind of protection that actually gives us against infection or disease or transmission or what have you, and if it's actually what the benefit is.
I also like to know when it's going to happen in the year, right? Because it's not clear to me that I mean, this has been going on all year. It's not just a winter disease, right? COVID.
Right.
So is it going to settle down and become winter, or because then you'd want to boost people then if you want to boost certain populations, right? But I'm not going to get an annual shot. I'm just going to stay with my original 3. I think it's fine. What's the dose timing for the second dose of Shingrix? I think it's 6 months recommended by CDC. And I wouldn't know without going back to the literature. I mean, I'm sure they didn't try different gaps. They just tried one and it worked, right?
Mm-hmm.
Thank you, Siddhartha, for your contribution. I think Siddhartha is coming to TWIV 1000. I recognize that name. I look forward to meeting you there. Okay, so Vanity says morning vaccine coupled with sufficient sleep is ideal, and there's the link for the study. All right. Thank you, Vanity. Brianne, what are you most excited about in immunology?
I would say that some of the coolest data that I've seen is about, memory and how different exposures can influence that. I think that some of the germinal center data from Gabriel Victoria is really exciting. I personally am very excited about kind of the way that the immune system and immunopathology and those inflammatory cytokines can be impacting pathogenesis. And so to me, the way that the immune response both protects from but also can cause pathogenesis is what I like to spend most of my time thinking about.
Okay, leukemia patients that get high-dose chemo followed by bone marrow stem cell donation lose all their vaccination benefits. Is there any way to donate immune memory to a naive immune system?
So in that case, the way you would do a donation is that you would be donating, trying to get some memory cells to donate. And I guess I could imagine ways that you could get memory cells to donate. But when we do bone marrow transplants in general, we do the stem cell transplant, as you say, and not the full bone marrow, because we don't want to transfer memory cells, because there's this complication called graft versus host disease, where those activated memory cells, if the MHC match isn't perfect, which it's really likely not going to be, those memory cells, because they're memory cells and are already activated, can start to attack the patients. So I worry about how you would donate immune memory without worrying about graft versus host disease.
Right. Okay, how does mental stress and physical stress affect and suppress the immune system?
I think that's all going through cortisol, or at least
Yeah.
a fair amount of it's going through cortisol.
Your thoughts about the Cuban vaccine that hasn't been on 3-phase trial? The one I know about has been. It's Abdallah, the protein subunit vaccine, went through phase 1, 2, 3, very effective. They immunized most of the population. I don't know one that didn't go through phase 3. Are you aware of any such thing, Brianne?
I'm not, no.
John, thank you for your contribution to science communication. Brianne, your knowledge has the breadth and recall of a good immune response. It's not shocking you did well on Jeopardy. All right. Peter says, office hours has been a resounding success. I hope biology with B and V will join life with D and V and Q&A with A and V. We're going to have all these. It's supposed to be life with V and D, to be a little provocative there with Dixon and Vincent.
Yes.
All right. Let's see. Dr. Casadeva was amazing, but his overall view of the sciences. Will Dr. Griffin be changing his treatment protocol to escalate serum? Well, I think if you listen, Casadeva didn't say it's for everyone. It isn't. It's just for immunosuppressed who do not respond to vaccines, and there you can treat them. So, it's he said that it's not for everyone. So I don't think Daniel's going to change anything. Brianne, did you ever want to be a physician, immunologist, allergist? Why or why not?
I did for a short period of time. I was pre-med for a very brief period of time. And I think that I had to do some patient advocacy work. And I realized how quickly a lot of the physicians were in and out with the patients, and weren't necessarily, spending long amounts of time, thinking about what was going on with any individual patient. And I kind of liked the idea of thinking about the same kind of issue or problem for many years. And I've just always been much more, I was much more excited about doing the science and the scientific process. Though, like I said, I did think about it for a very brief period of time.
David, thank you for your contribution to science communication. Thanks for helping me pass my immunology test. All right. Christina writes, is it possible that a person under extreme stress and their body is creating larger amounts of cortisol have larger chance of developing long COVID if only twice vaccinated?
Could be possible.
We don't have any data on that, right?
I don't think we have any data on that, and I think there's still a lot to be learned about long COVID and what exactly is going on there.
Do lymphocytes mainly tackle viral infection, or do they also play a role in fighting bacterial parasitic infections? Do neutrophils play a role in both viral and bacterial?
Lymphocytes absolutely play a role in viral infection, bacterial infection, parasitic infection. Lymphocytes are basically the cells of the adaptive immune system. They play a role in all sorts of types of infections. Neutrophils, I think, can play a role in viral and bacterial. I think they're thought of more in bacterial, but I know that they certainly can play a role with certain viruses.
Is sickness behavior due entirely to cytokine secretion being sensed by the brainstem hypothalamus?
There's a little bit of data on that, but there's a lot more data that we don't have. Some of that is data actually comes from C. elegans, who don't have a brainstem or hypothalamus, but they do have cytokine-like molecules impacting neurons. But I don't know if I could say that it's entirely due to any of those things.
Tia, thank you for your support of science education. We appreciate it. What's the association between vitamin D levels and COVID morbidity?
Oh, that's also a Daniel thing.
I don't know. It's a good question. I think it's very hard because so many people take vitamin D. It's hard to tease out from other things. So Elena is going. We got her deposit today, not deposit, payment.
All right. I'm excited to see you.
We'll be all there. We are going to have probably a full house, 130 people. It's really good. All right. Don't worry, be happy wants to know. What tests can be done to measure how good of an immune system you have? Would the results only be valid for a short time, or are they indicative of a more permanent state?
I would love to know the answer to that. Don't worry, be happy. Most of the things we can do right now are a sort of snapshot of a piece of your immune system, but I'm not sure if that tells you overall how good of an immune system you have. And immunologists love to talk about Goldilocks. Too much, too little is not good, but too much is also not good, leading to autoimmunity and other kinds of pathology. So I don't really know what a good immune system is. I think the results would probably be valid for a short period of time, but maybe some of those immune profiling studies are going to come up with an answer, and I will be right in line to find out the answer for myself too.
SRR, thank you for your contribution to science communication. Visto wants to know, could my dog get H5N1? He bites birds and eats their poo. I'm not aware of dogs getting H5N1.
Yeah, I'm not either. And in that case, would it have to be a GI infection, or would there be some aerosolization?
But it would be GI, yeah. So H5 in birds, influenza in birds is a GI infection, right? So
right.
It could be. So I have a paper here. Emerging infectious diseases, avian influenza susceptibility and receptors in dogs. Inoculation of H5N1 into beagles resulted in virus excretion and rapid seroconversion with no disease. How did they inoculate it? Because if you fed it to them, they would excrete it, right?
Yeah,
Here we
right,
go.
exactly.
To study, half the dose was given intranasally and the other half intratracheally. Okay, and then they excreted it.
All right.
So, yeah, the beagles at least get infected. They don't get sick. I don't know how you can stop your dog from eating bird poo, because they're going to be running around outside, right? Unless you keep them inside all the time. Okay. For your information, the bone marrow transplant with marrow mutation hasn't always cured HIV, so it isn't 100%. That's right.
That is true.
Brianne, do you think you could do a CRISPR approach, pull out stem cells, CRISPR out CCR5 and put them back in? Or do you think they're going to be off-target effects? That'll be a problem.
There might be off-target effects, and I think that could certainly help the lack of donors issue. But I still think that would be awfully hard for 35 million people.
Yes, that would be hard. There are not enough places to do that infusion. Yeah,
no.
You're right. Okay. On spillover, there have been novel deadly coronavirus every 10 years. Yeah. Took 10 years to find the cause of SARS-1. That's right. So we're going to have 10 years of this nonsense of people saying it came from a lab, right?
I don't know if I could listen to it for 10 years.
Do you think FMTs will become a more popular therapeutic? They seem very effective with C. diff. I've even heard of possible weight loss. Is there a downside? Fecal transplants, right?
Yeah. I mean, I think that there have been some great outcomes. I think there are some C. diff patients who seem refractory and don't seem to have a benefit. Yes, I have heard people using them for weight loss, but I've also heard people who have been trying to treat the C. diff and have ended up transferring other things, like, in fact, higher weight. So I think that there still is some information that we need to know, and FMTs are not foolproof.
Okay, what are some examples of PAMPs?
Oh, this one I got. So LPS, lipopolysaccharide, coming from the gram-negative cell wall, was sort of the first big PAMP that people described. Peptidoglycan from a prokaryotic cell wall, beta-glucan from fungal cells, different types of DNA and RNA that might come from viruses or other microbes. That's actually what I study in my lab. There are tons. Flagellin.
Okay, Carmen writes, in episode 977, I recall one of the reasons that led to immunosenescence is due to no longer having a naive immune system. If one was rarely to get infected, would one have a young immune system?
If one was rarely infected, I think that would also mean one didn't have much in the way of a microbiome.
Right.
And so I don't know how you would get few enough microbes in your life that you would ever have a young immune system.
977, was that the one where the retroviruses and senescence connection? I think so.
I think so. I'm not sure.
TWIV 977. Aging is infectious. Yep, that was it.
Yes, that's the one.
Thank you, Mike and Lori, for your support of science communication. We appreciate it. Okay. Is it possible for a person who is 70, got sick in February, I presume from COVID, possibly accelerate onset of frontal temporal lobe dementia? There's a family history of FTL dementia and Alzheimer's. I think if you asked Daniel this, he would say, we don't have enough data. He would say it's possible, but we don't have enough data.
I agree.
When is the FDA meeting next to discuss the COVID vaccine? I don't know. They just had a meeting, right? They just had a meeting in February, so I'm not. It's going to be a while, no? At least 6 months. There are unlucky people with a couple of Neanderthal genes with greater susceptibility to SARS-CoV-2 and others who never get infected. It's just our natural variability. We did that paper early on, remember?
Yes, absolutely. So it's certainly part of our natural variability. And there are a surprising number of immune genes that seem to be coming from the Neanderthal genome. That's interesting, and I don't really know anything about why that is or what else we could say about that.
TWIV 676, tragic gene flow from Neanderthals. Some people got mad that we were blaming Neanderthals on tragic gene flow. We weren't blaming anyone. It's just the way it is. I bought a TWIV cup with the virus on the front. Is that SARS-CoV-2? No, it's poliovirus with its receptor stuck on it, okay? Because that's the first structure and the only structure ever that we participated in. So polio is in magenta, and then the receptor is in blue. I mean, it makes it look like a virus with spikes, right? But it's not. I can see where you think it might be SARS-CoV-2, right? But it's poliovirus with its receptor. Steve Goff always used to say, you should change it. It looks ridiculous to have the receptor stuck on it. Well, we did that. My student made the protein. We're very proud of that. All right, Shingrix is 2 to 6 months. Okie dokie. What time of day is best for the shingles shot, the Shingrix? I have no idea.
I don't know. I don't think
Nope.
it was in the paper. They'll have to do another study.
Are immune memory cells, whoops, I didn't click it, are immune memory cells transferred in convalescent serum? Any risk of graft versus host there?
No, in convalescent serum, you are just transferring the serum is the liquid part of the blood without cells. So you would not have any immune memory cells transferred there.
Right. With respect to circadian patterns to immune activity, I thought several papers showed elevated brain surveillance via lymphatic system at night. Okay.
That sounds vaguely familiar to me, but it's a while ago since I've read about it.
When one receives blood transfusions, blood loss during surgery, what favorable immune traits might one gain?
So when you're getting blood transfusions, I mean, you're going to get some antibodies, I would assume. And so maybe you get a little bit of transient protection from something with antibodies. I don't know how often you're transplanting cells, particularly white blood cells, in those transfusions. So I'm not sure.
Peter, thank you for your contribution to science communications. Appreciate it. Steph asks, can you comment on the connection between immune system and the microbiome?
Sure, your immune system definitely impacts which microbes can colonize your body, but the microbes that colonize your body can definitely impact how your immune system develops and how it works. And so both of them are impacting the other. And to be honest, you can also actually throw the endocrine system in there too and make an even more complicated triangle.
WNPR mentioned you today here in Connecticut on Colin McEnroe's show. Yes, they emailed me yesterday to come on today, but I only saw the email this morning, so they didn't want me anymore.
They emailed me too, and I had a meeting.
Yeah, they do. You, me, and Alan are often on it. Okay, John says here, this question may be too late. Am I remembering correctly that H5N1 is developed and ready to go? There is one. The U.S. has a stockpile, but it's not clear how well it would match this currently circulating H5N1. And I remember the guy from Penn, what was his name?
Scott Hensley,
saying he could make an mRNA vaccine very quickly. They're working on it right now that would perfectly match it. So,
yep. I really like that conversation with him.
That would be the way to go, I think. Let's see. Okay, if we had copious funding, would it be useful to follow people's immune systems through days, weeks, months, years? I bet we could learn a great deal. What do you want to follow, though, right?
Yeah, but I mean, we'll do a whole profile. I would be fascinated. I'd want some data on how they were feeling, too.
Yeah, medical history would be good.
Yeah.
But you'd take blood and do everything you could possibly do? You want a lymph node
that
biopsy,
would be a start.
Don't you? You want a lymph node biopsy.
I don't want a lymph node biopsy, but that a lymph node biopsy would certainly give you even additional info.
Sure. I watched BARDA Industry Day on YouTube. The plan is to bridge with antivirals until full deployment. What bridge what? I don't know what you're talking about. Sorry. HPV equals PAMP, repetitive presentation of the L1 protein on the massive virion. It's not so massive, but it is repeated, yes.
It is. I think that the PAMP we usually think about from a papillomavirus is the DNA that's unintegrated in the cell. I have not seen papers on L1 as a PAMP, so I don't know.
German opera singer. How good is the chickenpox vaccine at presenting varicella zoster infection as opposed to symptomatic chickenpox? Would those who received the chickenpox vaccine in childhood need Shingrix? So I understand that they're testing Shingrix to replace the chickenpox vaccine
Right.
because it establishes latency, right? And you can
Mm-hmm.
get shingles at a low level even if you have
Right.
that.
And when you say it, you mean the chickenpox vaccine.
Yeah, the current, attenuated, infectious attenuated
Right.
chickenpox vaccine. So I don't know how good it is. It's better than getting the actual virus in terms of shingles, right? Which is what they mean by VZ infection. It's not
Right.
really an infection; it's a reactivation.
Right.
So, but you can still, at a low level, have reactivation after the attenuated vaccine, which is why they want to switch to Shingrix. Does that make
Yes.
sense?
I think that makes a lot of sense. I wish that I had the vaccine instead of having had chickenpox.
Did you get Shingrix yet? No, you're too young, right?
No, I have not had Shingrix yet.
Well, you can get it. I just got it this year or last year. I got it in the end of last year. But I had shingles also. Actually, the first year I started TWIV, I had shingles. So I remember that year, 2008, and I talked about it, I think, on episode 8. Can we use CRISPR to make human nucleated cells not susceptible and permissive to viruses like herpes?
I am not sure if I remember what the receptor is, but my guess is that there might be some downsides to this because some of those genes that we need that we would have to remove to stop susceptibility and permissivity are things that we would use for other purposes in our cells, is my guess.
I think there are too many cells you'd have to modify. I'm not sure you could get to them all with stem cells, or I think that's hard. Whereas with
I mean, you'd have to modify embryos, and that is a whole other can of worms. That
no, we're not going to do. That's unethical. We can't do that.
Yeah, exactly.
So I don't think that's happening. No. Oh, I'm sorry you broke your TWIV mug. Can you explain why some people get symptomatic respiratory diseases and others never seem to catch anything?
There's immune system variation, MHC variation in, SNPs in all sorts of immune genes and other aspects of immune experience and cross-reactive memory.
Yeah, also people may catch it but not get sick. They may have asymptomatic infections, right? So you just don't know. Peter says Neanderthals were coping with northern microbes for millions of years. I don't know if millions of years is right. They weren't around that long, were they?
I don't think so, but not up on my Neanderthals either. You guys are testing me tonight.
How long did they last? That's what they existed for, about 200,000 years longer than modern humans have. And we've lasted for 430,000 years, they think, not millions. Anyway, it's possible that H. sapiens made use of their genetic contributions, for sure. Absolutely. Because they did mate, apparently. With respect to those lucky people who never get infected, we're not sure where they didn't get infected, right, because they could have been asymptomatic, absolutely. What are danger signals?
So danger signals are kind of a term that was originally used to come up with the same idea or the same thoughts of PAMPs, that these are signals that tell your immune system there is a microbe around. And so whatever antigen they're seeing is dangerous and not just self. It induces some of those co-stimulatory molecules. You can also think of the DAMPs as being kind of danger-associated molecules. There's something killing our cells or damaging our cells.
All right, so Peter corrects himself. Neanderthals and us had a common ancestor hundreds of thousands ago. Okay, very good. And yes, Jeff Beck died of bacterial meningitis. Yeah, should we get the vaccine? I don't see. It's not a bad idea.
A lot of college students they recommend it for a lot of college students who are going to live in the dorms.
How is convalescent plasma obtained? From blood banks, and they, if they want COVID patients, they give you a questionnaire. Actually, if you give blood, they ask you, have you had COVID? And then it's put on the information for that unit. And then if someone calls for it, they know what to take. And it's not pooled. It's interesting. And so you get someone else's unit. And so Arturo said the best thing to do is because you don't know if they actually made antibodies, or because they just measure total antibodies, Brianne. He said that's a good correlation with whether you have neutralizing antibodies. But the best way to cover your bases is to give units from 2 different people.
Okay.
Is there a benefit to challenge trials? There can be.
There's a benefit in that we get some cool data, but does that benefit outweigh the potential cost?
Yeah, I mean, with SARS-CoV-2, you could kill people. You could give them long COVID, right? So
yeah,
you have
exactly.
to be very careful.
There are lots of interesting questions that we could answer, so that is a benefit. It's more of a cost-benefit analysis thing, and I don't know if it outweighs the cost.
So Lucky Six is meant bridge with antivirals in the event of a H5N1 pandemic until vaccines. Okay, because we have flu antivirals, and they work for H5N1. Very good. Got it. So SIP3 listened to 6 hours of the first TWIVs today. Wow. Good for you. I'm glad you like them. Those are interesting episodes.
Very cool.
Speaking of lymph node biopsy, can you get a supply and do research on lymph nodes taken during breast cancer surgery? Wouldn't they be discarded after pathology reviews? Well, if you had an arrangement with the surgeons and the pathologists, you could get them, but you'd have to have an arrangement with them. You have to have a protocol in place, and then you could get them, and they wouldn't throw them out. Yeah.
Right.
So, for example, in clinical laboratories, they screen people for viruses. Say they do nasopharyngeal swabs, and they say, okay, these are enterovirus positive. Then within a week, they throw them out because they can't store them all. There's no room.
Right.
And if you want them, you have to make an arrangement. So Amy and I were going to do a study, and they couldn't put the manpower aside to take the tubes and put them in a freezer for us, and we would get them. That's manpower that they couldn't supply. We had to somehow come up with money to either hire a part-time person or to reimburse them or something. It's very interesting, because it takes time. It's not just...
first lab. Yeah.
Go ahead.
The first lab that I worked in had a lot of projects related to the thymus, and all of the thymi that we had were coming from patients who had cardiac surgery, who had to have part of the thymus resected to get access to the heart. And so it was all the same kind of thing, that they had an arrangement so those thymus pieces could be kept.
Yep. Yeah, you have to have an arrangement, and it's a lot of paperwork and committees reviewing your protocols. It takes a long time. And in some cases, you need to pay for the time it takes, because in the case of the clinical lab, they can't just hand you the tubes. Things have to be done properly. I don't know did I ask, is coagulation part of innate immunity?
We are learning more and more about ways that it is, that some innate immune cells can make coagulation proteins and even throw clots. So that's something that I personally have been learning a lot more about in the past couple of years.
Thank you, Doreen, for your contribution. Glad you enjoyed office hours tonight. Susan wants to know if ATP is a DAMP.
Extracellular ATP can be a DAMP. So you can imagine it's coming from some cell that died and released the ATP out extracellularly.
Right. Yes, in the UK, they like challenge trials. That's where they did the SARS-CoV-2 challenge trial. We had a lot of problems with that on TWIV. I think that
we did.
Just was not a good idea. Claire, thank you for your contribution to science communication. And that's it for the questions tonight. Any other questions before we say goodbye to all of you and thanks to all of you? Well, I can start. Thanks, everybody, for coming. Let's see what we have. Oh, this is good. We have 386 people and 346 likes. That's good. That's probably the biggest office
Cool.
hours you ever had, Brianne, right?
That is definitely the biggest office hours we ever had. I do not have enough chairs. I do not have enough candy. I could never have had that many people in my office.
So thanks, everybody, for coming. Thank you to our moderators. Let's see. We had Tom, Les, Steph, Vanity Nutrition, and I saw PDK. I think I got everyone. Thank you so much for coming tonight. Brianne, thank you for joining me tonight. Hope you enjoyed it.
I did. Thanks for having me. This was a lot of fun. These were really great questions. I was very impressed. Kept me on my toes.
And I hope you come back sometime and we'll do it again.
Sure, absolutely.
Okay, folks, have a safe week. We will be back next week, the same time, Wednesday, 8 p.m. Eastern. Probably you will just have me next week. I'm working on getting another guest. Maybe that guest will come next week. But in the
Yeah,
meantime,
I'll be in Tucson visiting family.
don't sound so sad. It'll be warm, right?
Yeah, it will be warm, although apparently it's been colder than normal there recently.
So Vanity wants to know if you saw Acapella's leukocyte parody.
I did. It's so good.
And what's the painting behind you?
That is a Jackson Pollock. Um, yeah, the name is Convergence of
Cool.
that Jackson Pollock. Yeah.
Let's see. So should we get Trudy Ray? Okay, and Alan Dove should do one as well. That all works. We can do that. All right, folks. Have a safe week. See you next week. Good night, everybody.
Thanks, everyone.